Hello again, everyone. We're talking about generalizing our results this time. And when we speak to generalizing results, this is a broader topic than it seems. It really speaks to the representativeness of our studies, as well as the sort of meaningfulness of those results for various sorts of populations, or potentially the consumers of our research. So it's very important in our studies that we detail exactly who's in our studies, how we obtain them, and sort of the implications of those decisions for the general public and potentially researchers working in the same area. I like this image, and look away if this is bothering you, but this basically sort of represents in a couple different ways generalizability to me. First of all, just the idea that generalizability really spans out. The concept is the idea that our study has implications, or the results, reach a broader audience, like this spiral kind of implies. But then also that the collection of studies in an area, each individual study, like if you think of each individual color in the spiral, has some meaning to it, and the collection of those creates something different than the individual study does. So the larger picture is more important than the individual studies themselves. So we'll talk about a few of these elements today, how populations and samples sort of relate to this, and we've talked about this before, as well as threats and boundary conditions. We've discussed this with interactions and things like that that we'll go over a little bit, and describe generalization as an interaction between, let's say, people as the sort of dictating variable or the moderator, and we'll see how the IV-DV relationship changes depending on the types of people or how you look at those people. Consequences for internal and external validity, how lab and field studies differ in generalizability. In essence, lab studies maximize the internal validity, where they sacrifice some of the generalizability or external validity, and where field studies are capitalizing on that generalizability or external validity by being in the field with real people in real environments. However, you cannot control those studies as easily, so you sacrifice some of that internal validity, that real direct evidence for cause and effect. And we'll talk about a few different types of replications that are most common. So first of all, remember that populations, right? This is the people that we really want to ultimately generalize our results to, right? So this is humans or biological creatures. Remember, some research is really looking at the nature of biology, and that's why we can create those animal parallels through animal research. A lot of times it's just the psychology of humans in our field. So we assume a lot of humans work kind of the same way. And of course, not all of us do, and that's where those interactions and stuff come in social and personality psychology, right? But it may not just be humans, we may want to generalize specifically like Westerners, right? Men versus women. We may even look within sort of ethnic or racial categories to see different perceptions, let's say, of bias or something like that. But this is based on basically our sample characteristics, right? Whoever we use in our study is really creating those boundary conditions around who we should ultimately generalize our results to, right? So if we only want to generalize to women, then we can just include women in our study, but a variety of different types of women, young, old, different ethnic groups, things like that, right? The sample, right, that's the subset, right? So that's what we're talking about in terms of the people you utilized in your laboratory study, or to serve as the respondents for your surveys, right? So these should be as characteristic as possible to that larger population you hope to generalize to, right? And of course, any deviation there, any difference between that intended group that you hope to generalize to and the sample that you collect, those could be sort of those hurdles or barriers to generalizability, right? They may hinder your interpretation, we say, and the reasons for those differences between the sample population could be reasons that could create differences in the outcomes that we're looking at, right? Maybe income differences, socioeconomic status, stuff like that. If your sample's biased in that way, that could create a problem, right? And actually be one of the variables that could be most interesting. So, one easy illustration of this used earlier in the last presentation was just the idea that if you have this sort of population that you're trying to mimic, and you do not, let's say in this case, sample as many females as you should, then the processes may work differently, And of course, the generalizability may not be as strong if you have a different sort of people in that study versus the overall population, right? And gender, sex is an easy one to sort of see, but some things are a lot more subtle, right? Such as income. You may not see that as obviously, or maybe different intelligence levels or ability levels. So samples can differ on a lot of things, right? Culture is something we think about sometimes in terms of individualistic versus collectivistic types of cultures. This is important in a lot of research, let's say in industrial organizational psychology. Parenting sorts of styles also differ, right? So if you're doing developmental research and you're looking at children's abilities level or sleeping patterns or anything like that, those may differ dramatically by the types of parents these children have. Same thing as sort of even preferences. Different cultures have sort of attractiveness, sort of preferences that are cross-culturally valid. Some are not, and that's where that sort of cultural replication really comes in handy. And then, of course, your book talks about these weird samples, we call them Western, educated, industrialized, rich, democratic sorts of individuals who are usually found in our studies. It's in the 90-something%. Most research is done in the US and England and the Western European countries. So there's some in Germany too, and of course China has some research coming out of it, but a lot of it is Western educated, industrialized, right? So they're Western, they're highly educated typically, or beyond sort of, let's say, high school, industrialized in terms of there's, business and companies and all of that. And we all are above average wealth and more democratic than a lot of other nations. So the perceptions of these peoples and how they respond to things and their perceptions of the world may differ from someone, let's say, like my friend did Peace Corps in Madagascar, or individuals in Alaska, or even, let's say, in some of the countries we don't talk a lot about, let's say Finland. They have, sort of only slightly different political system, but they may have different cultural ideas about parenting and things like that. And of course, work within my field. One thing we like to do to sort of equate these groups is to look at pretests and somehow do matching. We talked about this as quasi-studies. Some investigations may need to sort of equate, let's say, one group of mothers to be as similar as possible to the other because you don't have, let's say, the randomization of a true experiment. So that matching procedure can help eliminate these confounds, and using these pretests and matching procedures can sort of help avoid some of these differences between groups, and in some cases help with this generalizability, though we can argue that it's more internally valid than not. We also talk a lot about college students as a potential source of bias, right? So a sample that's made completely of college students, but asking questions about work, let's say. My journals do not love that. They like to make sure that everybody in the study is a working adult, and sometimes they're just mostly got from college campuses, right? But the real question there is, would the process differ? Are college students seeing the work world or work motivations differently? And if that might be the case, then you probably should be sampling a wider age range and experience level of workers, right? Same things with rats or any sort of animal research. The more biological research is looking at things like, let's say, addiction. If sort of the addiction processes differ between rats and humans, then you shouldn't be using them to parallel the systems and investigate the sort of questions we have. But in many cases, there actually are very strong parallels to how addictive chemicals can react with our brains and our behavior. So in that way, it's important that we can study that in rats because we can't make children addicted to cocaine and stuff. So that's very helpful stuff. Anyways, volunteers can be just as problematic if we're looking at studies about helpfulness, anything like that. People can be more or less helpful, of course. So if you're asking for volunteers, they're already more helpful than the average Joe or Jane, right? And of course, situations matter. So I have a picture of a higher-end grocery store. Not only will then you maybe end up getting a certain income level of people, but they may also associate the context of when the survey or the questionnaire is being asked with the environment itself, right? So generalizing that to people shopping at other stores with other cues may not be accurate. Some of these things I've been referring to as threats or boundary conditions, right? So threats are threats to generalizability, your ability to say that the study matters for other people, right? So those are things like issues, right? Boundary conditions are sort of parentheses around, or they're a fence around who your research applies to. So let's say the phenomenons you see happen within a certain group, but they do not happen within another. So that the relationships just don't exist as strong, or something like that. For example, volunteerism is more of a threat, where gender-specific research is something where you can see that something is only important or, let's say, meaningful to, let's say, women, right? And you can't ask men about certain questions, or maybe certain relationship sort of dynamics are more important for women. So in that case, you would just do women in that sample and collect women, and leave men out of it. And maybe some other research, maybe recontextualize or try to replicate that with a male sample, but for different reasons maybe. Another example of that is how demographics can actually drive the results that you see, right? So they give some interesting studies in the book about this. One is more about cultural bias, but the same way can happen with demographics, where if you're looking at sort of this fish image, as they use in the book, North Americans versus Japanese individuals see different stuff, or notice different things with different backgrounds. And so you can see those patterns exist culturally. Same thing with demographics, that the gender of the participant or the sex can actually dictate the response to things more so than what you predict is actually at play there. So we describe this sometimes as an interaction, right? A statistical interaction. So generalization could be viewed as sort of a IV times moderator sort of interaction on the dependent variable, right? So these can be displayed as line graphs, or sometimes people use bar graphs for interactions, showing that the bars can be different, just like in the previous example with the Japanese and North American views on the fish, and how they see different or notice different things. In this case, let's say it's males versus females in some sort of environment where there's a different number of men present. I like this example with an elevator. So the different genders react to that stimulus differently, right? So if you say that the IV affects the DV in a certain way, that would be wrong or incorrect for different genders, right? So I think the interactional sort of way is the best way to think about it, because if you think that, let's say in this example, you get in a room or an elevator and there's one male present, everybody stands about the same height. The more men you introduce into that environment, the females tend to shrink, the men tend to grow, and actually physically get taller in terms of their percent of height. This is made up numbers here, but the phenomenon exists. So you could say here that gender is really sort of a gender-specific sort of reaction to the height, right? That men react in a specific way and females react in a completely different way. So this can be sort of a hint at the validity of the findings, or what we say the extent to which the findings are true for any group, right? So height does not increase for women, it does for men, right? Japanese people notice things about that fish differently than North Americans, right? So that the findings are actually different for different groups or subgroups of some population. So that really is the idea of validity, that the relationship between the IV and DV is more valid or less valid for certain individuals. This was the original idea behind meta-analysis. It was called validity generalization at the start, because the major idea here was just to see that, let's say in the case of training, that one type of training, let's say a lecture, works for various types of students or in various types of environments. We know that's not necessarily true, so we try to do a lecture, some sort of lab exercises, readings, exams, all sorts of things to try to like integrate the learning in different ways to try to get you to gather the material, right? So this all relates to sort of the two types of validity that we've been talking about all semester, the internal and the external. Remember, the internal aspects of the study that sort of add sort of evidence for cause and effect, or the external is that this sort of either replicates itself naturally in the real world, or that does relate to some sort of real world phenomenon or what happened within real world people, right, in their natural environments, in their homes or at work. We sometimes call this ecological validity, that it has some sort of real world parallel. So when we do stuff in the lab, as I said earlier, the lab, we have a lot of that internal validity, right? The validity of our research findings to real life might be more difficult when the distinction, however, between that lab and the actual field setting, that real home environment, that real work setting, isn't the same. So when the lab isn't like the field, that's really problematic for external validity, right? We may have really strong internal validity still, but that external validity might be really threatened. Field research, or research that's happening in a workplace or in a home, something like that, where it's natural, right? That capitalizes on all the benefits of the real people, real situations, all the sort of motivations and constraints within those natural environments, right? Present in that context naturally, right? The work environment, the family, sort of household where the work-family conflict is happening, but you lose a little bit of control, right? There's other stimuli present that could cloud the internal validity, but make that generalizability so high, right? So the relationship between sort of external validity and internal validity, is such that we want to maximize both if possible, right? Because when a study takes place in the real world, it occurs in a field setting, and it has the highest level of external validity, right? However, laboratory research can be just as realistic as research conducted in the real world if it has high experimental realism, right? So if there's some sort of realism present in that study, it can duplicate the field study and maybe have as much internal validity as possible, right? When you're in the field study, a field setting, right, you have a high external validity, so you really want to try to maximize that internal validity. So use good standardized measures, maybe the same data collection, research assistant each time. There's subtle things you can do to sort of maximize the consistency in that data collection. So the lab versus the field is a big debate here, right? With the experimental realism, we have more internal validity, right? So in the lab, more mundane realism is present if we have external validity, right? So the mundane is it feels like the real world. Experimental is when the lab duplicates that, right? The control versus fidelity issue is all about whether we can control all of those environmental cues and extra people in the environment, like we do in a lab study, versus the fidelity of that mundane realism, that everyday sort of feeling, right? Not just the experiment feels like the natural setting and that experimental realism, but mundane realism is really that it's an everyday occurrence, right? You want to maximize control, but also have that real-world fidelity, right? Unfortunately, effect sizes aren't so powerful in these real-world events, right? In the lab study, we have that control, so we can have larger effect sizes, but we have less of that fidelity. So there's sort of there's a balance there. More effect size in lab, but lower fidelity, right? However, in most cases, it's kind of determined for us, right? Some things like natural disasters, pandemics, are sort of uncontainable variables. You can't mimic that in a lab setting very easily. Things like, partner aggression, negative feedback, stuff like that, you can contain a little bit more in a lab setting and simulate it to some degree, having people watch videos and that sort of stuff. So some of the lab versus the field is sort of dictated for us. Some of it is a choice by researchers to say, I need the fidelity to be as high as possible, so I'm going to go into the field to collect this data, or I need the control as high as possible, so I need to keep it in the lab, and then maybe, send out surveys or do, some sort of more externally valid, more other sort of research later on, right? In this sort of race for generalizability, or this attempt to show that our research is important for other groups, we tend to replicate stuff, or try to show that these results hold again, right? If it's just a pure replication, we call that exact replication. It's following the measures and procedures exactly. Conceptual replications are generally where we're changing out a operationalization, so a manipulation of an independent variable, or changing out a measurement device for a dependent variable, maybe measuring something a little bit better to try to capture something in a different way. Sexism research has gone through this a little bit, because sexism sort of measures have evolved, and the most modern measure is called the sort of benevolent sexism inventory, where there's hostile and this sort of benevolent, it's the ambivalent sexism inventory. So it's the hostile and benevolent sort of traits that are sort of equally negative, but sort of different in nature, right? So using that new updated measure would be a conceptual replication to see if maybe this provocation for sexist behavior, changes with the benevolent versus hostile instead of the traditional measure of sexism, right? But it's exact replication is a study, conceptual replication is really a study, but literature reviews are also a type of replication, just done through a narrative sort of lens, right? So it's not always about data collection. The research you gather is a form of data collection. And meta-analyses are just taking that to another level. They're doing sort of the literature review, but then quantitatively synthesizing that. So that's a replication in a way, because it's testing the generalizability of these ideas. Literature reviews do that narratively. Meta-analyses do it through statistical analyses. And then there's a couple other sorts of replication tests that your book, I don't think, talks about, but tests of theory, testing theories like social exchange theory in a variety of different sort of maybe laboratory and field study sorts of ways, and then tests of generalizability in terms of maybe cross-cultural studies, gender-specific sorts of research, and seeing how genders react to different sorts of, let's say, interventions. There's a lot of specific types of replications outside of just exact and conceptual. Your book adds an extra one that's called conceptual with an addition, or exact with an addition. That just means it's a replication and you're changing it, right? I would consider that conceptual, but anyways, the need for these replications are more or less depending on the type of original study that was done, is important to note. So if it's a sort of case study or a very small survey, replicating those sorts of findings is pretty important. If it's a well-done experiment, needs replication, maybe more of the conceptual kind, to see how else we can get at this, manipulate this, whatever. Meta-analysis is the least likely, because it is sort of a replication, right? It's a test of generalizability, so you don't really need to replicate meta-analyses very often. Journals usually don't publish them, but maybe every 10 years on a specific topic. So case studies, small N studies, non-experimental, observational surveys, all of those are a little bit more in need for replication. Once you get to formal experiments, and especially meta-analyses, less likely to be replicated, right? But some other ones that I wanted to mention that I think are important, things like cross-cultural studies are very popular replications. You'll see so differences based on socio-ethno differences, even psychographic sorts of investigations in terms of how people with different sorts of psychologies react, let's say even to different medical drugs or things like that. Anyways, test validity within, like, let's say an IO context like me, we look to see if things like the GRE work on different populations. If they don't work on different populations, then is it really a valid test, right? And that's where that generalizability and validity really comes to a head. Currently, we're not even mandating the GRE in a lot of situations in a lot of schools for graduate school because that validity is in question. Also effectiveness, right? So going back to medical research, different sorts of biologies and psychologies respond to different sorts of medical treatments, whether that's physical sort of drugs to, improve kidney functioning, or something that's more psychotropic, something like an anxiety medication, things like that. And going back to training for a second, the transfer of training is really about the transfer of what you learn in a training environment to the actual work environment. So that is the idea of generalizability, or that that link between the lab and the field when you think about the training you get at any organization you work for. If what they do in the training room doesn't then replicate itself on the job, then it wasn't a very valid training, right? But if what you learn in that training then replicates itself on the job, you have similar sorts of experiences, and those learning tools that they gave you in the training actually come into play, then it's a perfect sort of valid training, right? So the effectiveness of that training is sort of judged as a generalizability sort of idea, right? Anyways, I hope this helps you kind of think about generalizability a little bit. Super important concept, and I hope that you integrate sort of these questions of generalizability into your papers when you write them. There's always sorts of limitations based on samples or all sorts of things that you could look at to see if your results may or may not hold for other groups.